So what if the blueprint of life wasn't just biology, but this incredibly intricate circuit that we could actually start to engineer? Today, we're going to dive into how scientists are moving beyond just reading life's code to actively writing it. And they're doing this by treating gene regulation not just as a science, but really as a powerful art form.
You know, this quote right here, it just nails the whole idea. We're not just talking about crunching numbers. It's this creative, almost intuitive process. It's about taking the messy, beautiful complexity of a living cell and translating it into a model that's, well, elegant and understandable.
Okay, so here's the game plan. First, we'll get into how this art actually works. Then we're going to roll up our sleeves and build two fundamental circuits from scratch, a switch and then a clock. After that, things get a little wild. We'll see how even the simplest rules can lead to chaos. And finally, we'll wrap up by looking at what all of this means for the future of engineering life itself.
So how in the world do you turn the beautiful chaos inside a cell into a blueprint you can actually work with? Well, the key is a super powerful tool called abstraction. It's really the art of figuring out which details are essential and what's just noise. It lets us see the simple underlying logic that's running the whole show.
The whole process follows this pretty clear four-step path. First up, you define your system, right? Who are the key players? Which genes are we looking at? Second, you draw it out. You represent their interactions visually, kind of like a circuit diagram. Third, you translate that picture into the language of math. You write the equations that describe those relationships. And finally, and this is where the magic really happens, you use computers to explore the system's dynamics. You ask, what kind of behaviors are going to pop out from these simple rules?
Now, the most crucial piece of the puzzle here is the feedback loop. This is the fundamental building block. Just think of it like a thermostat. Negative feedback keeps things nice and stable, right? Like maintaining the temperature in your house. Positive feedback, on the other hand, pushes things to an extreme. Think of a microphone squealing when it gets too close to a speaker. Just keep those two ideas in your head, stabilizing and amplifying, because they are the absolute key to building both the memory switch and the biological clock we're about to design.
All right, let's get our hands dirty with our first creation, a biological switch. The whole goal here is to build a circuit that can sit in one of two stable states, either on or off, and hold that state, basically like a tiny little memory chip.
The design for this is just beautifully simple. It's called a toggle switch, and it's built from just two genes that are constantly shutting each other down. Gene A makes a protein that turns gene B off, and at the same time, gene B makes a protein that turns gene A off. It's this perfect molecular standoff, and it creates two and only two possible outcomes.
And this slide, wow, it just illustrates the result perfectly from a real experiment. See, each one of those dots is a cell. Notice on the left, the whole population is clustered in one state. Let's just call it off. But then, after they get a little temporary chemical signal, look what happens on the right. The entire population has flipped or toggled to a new on state. And they stay there even after the signal is gone. That's bistability, two stable states. It's a simple but incredibly powerful form of cellular memory.
What's just so remarkable is how cleanly this biological switch actually works. This graph shows it all. It has this nearly ideal threshold. You can push the system with a weak signal and nothing happens. You push a little more, still nothing. But once you cross that one specific point, bam, the system just decisively flips over to its on state. This isn't some gradual dimmer switch, it's a crisp, clean, digital switch built entirely out of biological parts.
Okay, so we've built memory. That's pretty cool. Now let's move on to our next design and see how just a simple change in the wiring can create a totally different function, a biological clock.
This little circuit here is called the repressilator. Now, instead of two genes fighting each other, we've got three genes in a circle of repression. So A shuts down B, B shuts down C, and then C comes all the way back around and shuts down A. It's basically a continuous chase.
And because of this chase, the system can never, ever find a stable state to just rest in. As gene A's protein level goes up, it shuts down gene B. But with B out of the picture, gene C can turn on. Then C's protein builds up and shuts down A, which then allows B to turn back on, starting the whole cycle all over again. The result? This beautiful, sustained oscillation. The cell now literally has a pulse. So far, we've created order, a stable switch, and a rhythmic clock. But you know, life also operates right on the edge of chaos. So what happens when we take these simple mathematical rules and just push them to their limits?
Now let's look at this deceptively simple equation. And don't worry about the math itself. This is the logistic map. and it just describes something really basic: population growth in an environment where there's a limit to resources. But, as the ecologist Robert May discovered, hidden inside this simple formula is a whole universe of surprising complexity.
So if the growth rate is low, the system is, well, it's boringly predictable. Imagine this line here is the population over time. It just quickly settles on a single, stable value, what we call a fixed point, and it just stays there. Perfect, predictable order. But now, let's start to crank up that growth rate. Suddenly, that single stable point just vanishes. The system can't settle anymore. Instead, it starts jumping between two distinct values, a big population one year, a small one the next. This first split is called a bifurcation, and it's our very first step away from simple order.
And this is where things get truly mind-bending. As we crank up that growth rate even more, the system splits again, now oscillating between 4 values, then 8, then 16, in a cascade that just gets faster and faster until, boom, it explodes into chaos. This visual mess you see on the right looks completely random, but it is generated by that exact same simple deterministic equation. This is the beautiful, predictable road from order straight into chaos.
So we've gone from these really simple circuits all the way to profound complexity. What does being able to understand and actually build these things mean for the future?
Well, the implications are just huge, and this is so much more than a theoretical exercise. On one hand, this whole approach helps us deconstruct the really complex gene networks that are already running in nature. But even more exciting, it gives us a toolkit to design totally new functions: cells with memory for biocomputing, programmable circuits for smarter therapies, and just countless other innovations in biotechnology.
So it all comes back to this one simple thing. By mastering the art of modeling these simple biological circuits, we gain the power not just to read life's existing code, but to start writing our own.
And that leaves us with a fascinating and maybe a slightly unnerving question. We are really transitioning from being just readers of the genetic code to its writers. So if life truly is a programmable circuit, well, the only limit is our imagination. So what are we going to design next?